Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality-assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers. Everything you need to learn, review and prepare for your SQA exams. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. You can now also collect Young Scot reward points by using the codes within the description of each episode to claim discounts and rewards on their website www.young.scot.
The Highlands of Scotland have a bit of a monster problem. There's at least 13.
DNA is the blueprint for life and is a double-stranded helix made up of repeating units called nucleotides. In this lesson, I'll be speaking with Professor Neil Gemmell from New Zealand on how he is using DNA, PCR and genomic sequencing to search for the Loch Ness Monster. We will discuss why he travelled across the world in search for Nessie and develop our understanding of some of the key areas of the higher DNA and the genome unit. We will discover some of the life that exists within Loch Ness and whether or not that includes a potential monster. You can subscribe to the podcast on your favourite platform to ensure you don't miss out on any episodes, or follow at Biology Higher on Twitter for regular updates. Loch Ness is a Scottish loch filled with mystery and legend. In 565 AD, St Columba travelled north in a bid to convert what he considered to be heathen Picts to Christianity, and according to his biographer, St Adamnan came across a monster in Loch Ness. To date, there have been 1,120 sightings recorded of the Loch Ness Monster, with 8 sightings this year in 2020 and 18 in 2019. Various hunts of differing sizes and budgets have taken place over the years, including a well-known Hollywood actor in 2013 who apparently used a large hook, some rope, a leg of lamb and malt whisky, but failed to capture the elusive and legendary creature. The most famous photograph is probably the surgeon's photograph, which was taken by a London-based gynaecologist called Lieutenant Colonel Robert Wilson. I guess the papers wouldn't have wanted to name the image the gynaecologist's photograph. The circumstances around the photograph seem to be a bit hazy, but the story goes that Wilson was travelling to Inverness with his mistress, and it was taken on that journey in April 1934. The photograph featured in national newspapers and most people would recognise it. It's been called a hoax, and the stories claiming it to be a hoax have also been called a hoax, but nonetheless the picture continues to be shared to this day, with people suggesting repeatedly over the decades that Nessie is some form of plesiosaur. Whether a prehistoric animal, an elaborate hoax or a simple trick of light, Professor Neil Gemmell wasn't afraid to travel across the globe to see how he could use his knowledge of DNA to find out more. Neil, nice to see you.
Nice to see you too, Paul. Thanks for having me on.
And maybe you could tell us a little bit about yourself.
Yeah, sure. So I'm a professor of genetics at the University of Otago, founded by Scots settlers actually about 150 years ago. And I have for a long time been interested in using DNA to answer questions in an ecological or evolutionary frame. I've done work on things like platypuses in Australia and worked on fur seals and various other species over decades now because I'm starting to get a bit older. And a few years ago, we started getting interested in this new tool called environmental DNA, which is effectively all the DNA that organisms leave behind as they pass through their environments, whether it be in water, air, or in soil. And starting probably about 6 or 7 years ago, we started using that tool in New Zealand to look at the biodiversity, the living creatures in our environment, particularly in the oceans, to try to get a shopping list, if you like, a census of life that occurred in those environments. And the real driving force for that was so we could better understand changes over time. So if we know what species are present, for example, in our harbour here in Dunedin, in 2015, for example, then we can look to see year by year how those species abundances and distributions alter. And that can give us some indication of whether we're improving our environment or whether we're degrading it further. And that was sort of our primary motivation. But that's not why I'm on the show or on the podcast. The reason I'm on the podcast is because starting about 3 years ago, we started asking the question, could we use this new tool to go and look for the world's hide-and-seek champion, which is the Loch Ness Monster, or Nessie. And really what we wanted to do there is to showcase the power of environmental DNA for understanding our environment better. I'll be honest and say I don't really believe that there's a monster in Loch Ness, but I've always been. I know, Scottish tourism will string me up, but there's been so many sightings and it's such a compelling story. We thought, let's go and have a look and let's see what we can find out. And as any good scientist should be, you start with a hypothesis. Now, hypothesis is that there probably wasn't anything there, but we were happy with the notion that perhaps we would be wrong. So we went out in 2018 and sampled extensively across Loch Ness looking for environmental DNA. And we'll tell you more about that as we go.
Yeah, let's start at the beginning then. So we obviously understand that DNA is the blueprint for life. It's a double-stranded helix. It's made up of repeating units called nucleotides. And each nucleotide has 3 chemical parts, a base, a phosphate group, and a deoxyribose sugar. There's 4 possible bases, and those linked together by weak hydrogen bonds, A always with T, C always with G. What do we mean, though, exactly when we talk about environmental DNA? You said it was obviously fragments that were left behind.
Yeah, so that's a very good question. It's pretty straightforward, really. So as I sit here in my lounge at home, I am constantly losing small pieces of skin or eyelashes or, the odd hair. And of course, in the environment, if I was an animal, I'd be defecating and be leaving traces of myself in that way, or I might be peeing, or what have you. So it's, if you like, it's the detritus of life is being captured by environmental DNA, and each one of those cells, whether it be a skin cell or a intestinal cell that's been caught up in a poo, that has inside it the entire genetic blueprint for that animal or plant. Each cell has the genetic blueprint inside it, and we can extract that DNA from those cells from an environmental sample, whether it be soil or water, or even in some cases air. So you can filter the air, and we find that there are tiny little particles of mostly bacteria and viruses, the tiny little particles of life, if you like, that we can track and trace. And obviously the amounts of material that are present within a single cell are tiny, but the techniques that we now have at our disposal enable us to make multiple copies of that so that we can zero in and analyze particular parts of that DNA or parts of that genome that might be present in a single cell. And occasionally the cells fracture and they break open, and you can actually get the contents, the DNA and the RNA that are present within the cell in the environmental sample also, although the stability of that is much lower than when it's intact in the cell. In fairness, this technique is very temporal. So what I mean by that is if I pass through my garden, the signal of that passage may last for a day or two, right, through a particular track. And so if I sample in the places where I've been within a reasonable time frame, I'll detect my presence or the presence of any other animals that might have been there. If I go back a week later, that signal will be much, much lower. But it's incredibly powerful, and there's some really cool science that's coming out in environmental DNA circles. There's people tracking polar bears using the DNA that is caught in their footprints in the snow, which I think that's pretty cool. And there's obviously our sort of work, which is looking at the biodiversity that's contained within, say, a litre of water. There's other people who are looking at sediments, and sediments are much more stable, and the DNA can give us a longer signal of the history of a particular environment. And there's people I know here in New Zealand and elsewhere who are drilling down into the sediments quite deep, meters into them, and that's going back thousands of years, and they're managing to extract DNA from those sediments and then reconstruct what life looked like in particular lakes, say, 100 or 1,000 years ago, which again I think is pretty incredible. And that's sort of a crossover between two technologies, one called environmental DNA, another one called ancient DNA, and they're very similar in many ways. Both fundamentally require you to extract the DNA from an environmental sample, and then you would make many copies of it, and then you would sequence it, analyze it against databases. So exciting times.
And of course, when we talk about animals and plants, we group those as eukaryotes, as having a true membrane-bound nucleus. And the bacteria that you mentioned, those would obviously, they would not have a true membrane-bound nucleus and therefore be prokaryotes. We understand at higher that within eukaryotes, that circular chromosomes are located in mitochondria and chloroplasts, and then we get linear chromosomes within the nucleus. Does that come into play with the environmental DNA?
Yeah, that's a really good question. So we predominantly have been working on mitochondrial or chloroplast markers because those circular molecules tend to be more stable than the linear chromosomes. They're also present in higher copy numbers in each cell, so that's quite handy. For bacteria, they're relatively robust. And in fact, actually, that's a really important point and distinction. So when I've been talking mostly about eukaryotes and particularly vertebrates and things that most people are interested in, particularly if you're interested in things like monsters, the issue is that you are collecting parts of those animals, as I say, the detritus of their lives. But of course, for some organisms, in fact, actually a very large part of the tree of life, we're collecting the entire organism. We're collecting the entire cell, whether it be a bacterial cell or whether it be a viral particle or whether it be a single cellular organism, which might be things like some of the lower eukaryotes, or they're multicellular, but they're very small and microscopic. Your nematodes and your rotifers and those sorts of things. And just quick spoiler alert here. Loch Ness is chock full of those things, and it's taken us a long time to work through the species lists or the taxa lists for things like rotifers and nematodes and a whole swag of other things.
You talk quite a bit there about creating copies of the DNA. I guess when you're talking about that, you're talking about processes like polymerase chain reactions.
I am indeed, Paul. That's exactly right. Effectively, what we're doing with the polymerase chain reaction is we're making copies of a particular target sequence within the DNA. So we're using what we call primers, which are short pieces of DNA, which are binding to specific regions in the genome, whether it be the mitochondrial genome or the chloroplast genome or the nuclear genome. And then we are using DNA polymerase to make a copy from those priming sites. And you are using a process, so the polymerase chain reactions, power comes from the fact that you are denaturing the DNA, you're annealing these primers to it. They bind specifically against the sequences that they match. The polymerase makes a new copy, and then you can repeat that process once the DNA is created by denaturing it again to make it single-stranded, and then adding in new primers, new polymerase, and the polymerase will make, and the primers will rebind in a new cycle and make more copies. I guess the analogy that's been used now for more than, I guess we're getting close to 30 years since the polymerase chain reaction became a tool which was widely used. People still talk about the idea of polymerase chain reaction being a bit like a photocopier. You're putting in one template, one page of paper, and you can make many, many, many copies of it in relatively short times. And it's an exponential process because you've got the primers in there, and you make 2 copies in the first round of the reaction if you've got one molecule, and then 4, and then 8, and then 16 as you go through what we call cycles of the reaction. Actually, things are moving on. So PCR, which is a very common technique in most molecular laboratories, and in fact probably in many high schools now, students will be doing it, and I think this is a good thing. It's nice to feel that you're working with the technologies of the day, and polymerase chain reaction still is one of the big backbones of modern genetics, modern molecular biology. But increasingly, people are starting to get into new technologies which enable detections without that thermocycling. So there's some clever ways to unravel DNA and then make it copy itself without having to go through the denaturation process. So anyway, that's probably a little bit too technical, but
one of the things that we do need to know for the higher exam is that thermocycling process and the different temperatures that obviously it works to. And obviously, as you said, the kind of PCR process starts by mixing those specific target sequences that are to be amplified with the free DNA nucleotide with some heat-tolerant DNA polymerase. And then those DNA strands get separated by heating up to somewhere between 92 and 98 degrees centigrade before the DNA gets cooled down to 50, 65, somewhere in between there degrees. Yeah. And that obviously allows those complementary primers to bind to the target sequences. And obviously DNA, of course, is double-stranded, so we need 2 complementary primers, one for each strand. And then after that, the DNA is heated back up to somewhere between 70 and 80 degrees for that heat-tolerant DNA polymerase to replicate the target sequence. So I guess one way of remembering it sometimes is that kind of heat, cool, heat, repeat process. It obviously gets quicker as it goes, but you can probably get up to, 30 cycles an hour?
Yeah, probably. It depends on the machine that you've got and how quickly it can change temperature. But you're right. I mean, so we would normally denature at about 95 degrees Celsius. It's sort of classic for us. And some of the enzymes that are used in the process require that heat to activate. So that helps with the specificity of the enzymes. They're called hot start polymerases, if you want to. And then what would happen is after the initial denaturation, then you've got what we call the annealing step. So that's when the primers are binding to the single-stranded DNA. And then we've got, and that's generally, as you say, around about that 50, 60 degrees, maybe 65 degrees, depending on the specificity of the primers. And then you go into an extension step, which is generally at about 72 degrees. So it's, and you're talking seconds at each of those stages, so 15, 30 seconds. And then the annealing step is generally a little bit longer, depending on the product size. There are lots and lots of rules of thumb on this, and many different labs operate in slightly different ways how they do their PCRs, but that's fundamentally it.
And of course, you do have to be back in the lab before you do the PCR. Can you tell us a little bit about how what equipment you were using to collect those DNA samples?
Sure, but before I segue to that, I'll just say that increasingly we want to be able to get this technique into the field so that we can actually do the extractions and the detections on site. But that's another story, perhaps for another time. So collecting the samples is relatively easy. I mean, so when we're dealing with Loch Ness, obviously what we wanted to do was to sample quite extensively around the loch. And of course, what makes Loch Ness particularly mysterious is that it is dark and it is also deep. So it's over 220 metres deep at its deepest point. It's probably about 229 metres. And there are 2 major basins of the southern and the northern basin. And so we wanted to get really deep samples of fresh water from each of those sites. We wanted to get sites from around Loch Ness near the shore, and we also wanted to run a transect of varying depths right through the centre of the loch. And remembering that the loch is about 21 miles long, so if we took for example, a sample every mile or so as we went up that loch. That was sort of our plan. We ended up taking something in the vicinity of about 150 samples from Loch Ness, and we also collected samples from some nearby lochs. The argument being that there's a monster in Loch Ness, but there's not in these others. Although, as it turns out, the Highlands of Scotland have a bit of a monster problem. There's at least 13.
So this is a new thing even to me, Neil. I mean, we all have heard about Nessie and the Loch Ness Monster, but Nessie's actually got a cousin further south called Loamy in Loch Lomond, which I have to say I've never heard of.
Yeah, the Loch Lomond monster's not one I'd encountered until I started reading into this. I'd heard of Morag, which is in Loch Mora, which is the deepest loch in Scotland. There's also Wee Oiki, which is in Loch Oik, which is connected to Loch Ness by the Caledonian Canal. Is that right?
The Caledonian Canal basically connects the North Sea to the Atlantic Ocean.
Oh, okay. Is that the one that goes west-east then? Must be anyway, the series of canals that connect the Scottish lochs with Ness and the River Ness being one of the main entry points into the North Sea. And then you go down through Loch Ness, and then I'm now going to show my geographical ignorance, but you've got Loch Oik, I think, and then you've got anyway, there's a series of them. And so you've got Wee Oiki, you've got Lochy, you've got Nessie, are all sort of they're next door neighbours basically, apparently.
Socially distant.
Well, I hope they're socially distancing now. But the interesting thing is that just trying to find a control site, a site where we expected to find no monster DNA, because there weren't meant to be any monsters there. There'd been no sightings. Actually proved to be a bit of a challenge. And also, of course, we'd like to try to match these sites so that they were of similar sorts of, so that the lochs were similar, and that proved to be very challenging too. So there are some lochs that are quite dark and peaty, so you could go and sample those. There are very few that are as deep as Loch Ness, so Loch Moira would be one of the few. So we had to make some compromises, but we collected about 50 samples from some other lochs too, just to see how they might differ, or more particularly how Loch Ness might differ from it. So when we're collecting, what we're collecting is just water. I mean, it's really couldn't be simpler. I mean, it's literally bucket science, although the bucket's a little bit more sophisticated than your household bucket, although not always. So we had quite a run on the supplies of plasticware in Inverness when we were visiting. So we went to one of the supply depots and basically bought out every plastic container they had. But we're collecting water samples, and we're using actually a quite a sophisticated tool, which was called a Friedinger water sampler. These have been around for hundreds of years. Basically, they are hollow tubes that have lids, has a lid and a bottom. And the nice thing about those is that you can get them to close when they reach a particular depth. And it's, again, not rocket science how you do that. So what we had was a rope which was marked out in sort of metre lengths, so we would know how far down we had put this device, and we also had a recording device on the bottom of it so that we knew how deep it had got. And when we thought it was at, say, 50 metres, we would send down a weight, and that weight would interact with a switch on the device, and that would close it all up. And then so it's capturing a litre or 2 litres of water or 5 litres of water, in the instance of some of the devices we were using, at a specific depth, at a specific time. And then we'd pull that back up, and then using all sorts of sterile gear, or keeping things as sterile as we could, we collect that water, and then we filter it to concentrate the organic material that is present within it. And then you repeat that process, 150 times at different depths and in different locations. So it took us some weeks to collect all the samples, and then longer to process them. But we're filtering them just through very, very fine filters, the sorts of things that you'd use in a laboratory to make culture media sterile, is what we would use routinely. So these have what are called Stereovex filters or the like, and the filters themselves have a pore size of about 0.2 microns, which is really small. and they're going to collect basically everything that's in the water, barring viruses, which will pass through. So we had concentrated the materials from bacteria upwards into a sample, which we could then take to a laboratory to extract the DNA from.
So you're not having to go through customs at the airport back to New Zealand with litres and litres of water?
No, and in fact, actually, no samples came back to New Zealand. So we have very strict biosecurity laws in New Zealand. And so the notion of us bringing some water samples would have been completely off, and the idea of us bringing anything that could potentially affect the freshwater systems of New Zealand would have been considered to be quite bad. So actually, all the DNA extractions were done in England at the University of Hull, and then we took those samples to France to do the polymerase chain reaction in a laboratory of a chap called Pierre Taberlet, who was one of the originators of environmental DNA technologies. And so we then did, I think, in a space of 3 or 4 days with Pierre, about 26,000 PCRs to try to look at the life that was present in the samples that we'd collected. And what we were doing there is we were using a process called metabarcoding. So that sounds very flash, but it's not particularly complicated. What we had were PCR regions. We were making PCR regions that were very good for describing particular parts of the tree of life. So there are PCR primers that you can get, which will pick up basically vertebrates. So birds and fish and mammals and amphibians and reptiles. And so we had a series of primers that were targeted at that because our leading guess was that Nessie might be some sort of giant fish. Obviously, some people think it's a plesiosaur, which is now extinct, but also a vertebrate. So, we should be able to amplify its DNA if it was a vertebrate and look for it. And we also had 2 sets of primers which were eukaryotic generalists. So they would pick up everything from fungi through to mammals and plants and so forth. Then we had another set which was specifically for plants because we're kind of interested in the diversity of plants. And then we had one set which was generic for bacteria, which would pick up most of the bacterial species that would be present in an environment. And so those were the targets, and we made multiple PCRs from each sample. So one of the biggest problems with the polymerase chain reactions, but its strength, is its sensitivity. So one of the things we have to be very careful of, or at least mindful of, is contamination. As obviously some of the primers that I was working on, the vertebrate and the eukaryotic primers in particular, were very likely to amplify human DNA. In fact, we knew it could. So of course, all the operators could be potentially found in the DNA signal from Loch Ness, either because of the way we handled the samples or because of contamination later in the laboratory. So generally what we were doing is we're doing 3 and sometimes 4 individual PCRs on each sample, and we only believed things if we found the same signal across all 4 samples. And we had controls, blank controls, that shouldn't have amplified anything. And if they did, then that suggested there was contamination through the process. And as it turns out, that when you do 26,000 PCRs, you do get contamination. It's just one of those things. You try to control it as best you can. And if you can't control it in the laboratory, then the thing you can do is you can say, look, here's my blank controls. Anything I find in these, I'm going to assume as contamination. And I'm going to take those out of my analysis downstream using bioinformatic approaches. And so that's quite a stringent thing to do. And you lose quite a lot of data in doing it, but that's sort of how we approached it. So we've got multiple replicate samples at each site. We've got multiple replicates of PCRs from each sample in the laboratory, and we're only believing things where we get multiple hits across the samples and the PCRs.
That then leads us on to genomic sequencing, which of course is the analysis of those sequences of bases within the organism's DNA. And as you said, that obviously involves bioinformatics, which I guess is basically the analysis of that data using computers and statistics.
Yep. Absolutely.
We tend to obviously look at things like the human genome a little bit, which
Mm-hmm.
I think consists roughly of, you can correct me if I'm wrong, Neil, but I think roughly 3.2 billion base pairs. And I think that was completed in April 2003. But since then, we've gone on and sequenced many, many genomes from since then. And I guess it's worth pointing out as well that the size of that genome doesn't necessarily correlate with the complexity of the organism either. I think the marbled lungfish has something crazy like 132 billion base pairs possibly, and certainly not as complex as ourselves. I guess all that sequence data and genomic data is held in a big database somewhere that you then cross-reference what's in your samples with.
That's exactly right. So there are about 3 big databases globally, and they mirror each other. In other words, so if I put something into the US database, then a copy of that sequence would be sent automatically to Japan, which has a database, and there's another one in Europe. And these are enormous now, and because the scale of sequencing that is undertaken compared to where we were at, say, 30 years ago is quite extraordinary. So I did my PhD sequencing most of a molecule called most of the mitochondrial DNA of the platypus. Was one part of my PhD, and that's 16,000 base pairs. And that took me better part of several years. And now that's the sort of thing that our lab would knock off in an afternoon. So the scale of science has grown astronomically. And so genetics used to be very much a laboratory-based discipline, and it still is to some degree, but you spend, my students and staff now spend most of their time analysing data. So they generate the data, and that might take them some period of time, and then they spend the rest of their time analysing it. And so when I did my PhD, I think I spent probably 90% of my time generating data and 10% analysing it. These days, the science is the other way around. So what we're doing with the sequencing, so we're using, and that's probably a misnomer now, it's still called next generation sequencing, although it's been around since about 2006, 2007. So 13 years. So for most of the students who are on this podcast, this is the sequencing that has been around for their lives. And this new sequencing is very fast, and it deals mostly with relatively short fragments, and you can sequence literally hundreds of human genomes on some of these high-capacity machines now within a few days. Very, very powerful technology, lots and lots of information. And the technology behind that, I think, is probably a topic for another conversation. But fundamentally, it's been driven by the fact that we can label individual nucleotides with particular fluorescent dyes. We can use lasers or and high-definition cameras to capture those light signals, and then we can build a sense of the sequence based on the detection of those light signals sequentially as bases are built in real time on a sequencing platform. So that's one of the technologies. And then there's some new technologies which effectively are pulling whole fragments of DNA through tiny pores or through particular channels and then reading them base by base, which is really exciting. And some of that's being led out of the UK by a company called Oxford Nanopore. So there's lots of different ways now to sequence things. They all have their strengths and limitations. We used what's referred to as short read sequencing, which has been around, as I said, for about 13 or 14 years. And the power of that is that you get many, many sequences for each thing that you've put into the sequencer. And that depth of sequencing can give you some level of accuracy in terms of the results that you obtain. And generally, your sequencing may be 250 base pairs of sequence at a time. And then you're taking that sequence and you're comparing it, as Paul said, to databases. And these databases contain all the sequences that scientists around the world have contributed for their particular species or their particular question. And so while we may not actually have the exact species that we find in Loch Ness, for example, we may find something that is relatively closely related to it. And we can say, oh, okay, that's a bacteria of this particular group, or that's an amphibian, and it's probably a common toad. Those sorts of things. Or this is a sequence that, based on our analysis and understanding, is unlike anything that we've seen before, but it probably fits somewhere between, for example, crocodilians and turtles, and that would be the sorts of thing that we would have expected if we had found a plesiosaur sequence, but we didn't. I know.
The tourist industry is hating this podcast. I guess at a higher level, we would talk about the comparison of genomes helping us to identify common genes between organisms, and also helping identify genes that make organisms unique. And that's obviously exactly kind of what you've just been describing, Neil, isn't it?
Yeah. So, those metabarcodes that we have targeted are regions of DNA that are highly conserved across parts of the tree of life. So all vertebrates will have it. All eukaryotes will have another one of these barcodes, and all bacteria, or most bacteria, have another one. Generally, for something to be conserved like that, it has to have quite an important function. So the diversity that you see, the amount of sequence change that you see in those sequences is generally quite small within a species, but between species it can get quite large. And we can use that as a very powerful tool to reconstruct the relationships amongst organisms. We weren't really doing that here, but we are using the power of this of these sequences as detection tools, things that we knew we should be able to, if there were vertebrates in the loch, and there are, then we would find those sequences.
So what sequences did you find in Loch Ness, Neil?
Yes, that's a good question. So it's quite a shopping list. And of course, what we have to do is we break it down basically by each of the primer sequences, or each of the metabarcodes that we looked at. So already, I guess last year, we put up what we thought were probably the results that people cared about most, which was the vertebrate set, which in actual fact is probably not the most powerful data set that we had. But the vertebrates were what told the story that most people were interested in, which was, is there any evidence that we found anything extraordinary in Loch Ness, any unusual vertebrates? And the answer is really no. We find all the fish species that we knew occurred in Loch Ness, and actually a couple of others that people thought might be there, but we weren't 100% sure. So, for example, stone loach and grayling come up in some of our samples in Loch Ness, although very rarely, and there've been some suggestions that they were there. But we find trout, and we find Arctic char, and we find salmon, and we find lamprey. And what we found was an extraordinary quantity of eel DNA. So either when we sampled, there was a big eel migration or something, and there were just eels all over the place. But many of the sites that we sampled had quite a lot of eel DNA in them. And so we sort of landed on the idea that if there was any sort of monster in Loch Ness, the only one that we could find support for was this notion
of a giant eel,
yeah. So we went looking for giant catfish, we went looking for sturgeon. We didn't find any evidence of those.
Was there any evidence of catfish, Neil?
No, we didn't. But I think one important point is to be, as a scientist, is to be honest about the limitations of any study. And so what I can say quite definitively is that we can't rule out that those species are there. We just didn't find them. And so it becomes a sort of sampling problem. So 150 samples sounds relatively impressive until you consider the size of Loch Ness, which at 21 miles long and about a mile and a bit wide and 220 metres deep, has as much water as all the other lakes and rivers in England and Wales combined. So Scotland's probably got a bit of a water monopoly, something to bear in mind as your neighbours to the south ask for it. Water, the new gold. Anyway, it's really big. And so the chances that we could have missed something are reasonably large, although if it was a very, very large organism, perhaps not.
The reason why I asked about the catfish was there was a recent photograph emerged of a giant catfish that supposedly from Loch Ness. But my understanding is that the markings on the back of a catfish are almost like a fingerprint. and are unique to that individual. And somebody had very carefully taken a look at this and pointed out that it was exactly the same markings as another giant catfish from another country elsewhere, and had been photoshopped in.
Yes, I saw that too. I don't know if they're unique, but it would be unusual, I think, for the image that appeared in Loch Ness to have been photographically identical to another one taken by a fisherman, another fish elsewhere. So yes, I think that one was, as they call it, a genuine fake. But nonetheless, there'll be people who want to believe. But yeah, just going back to it, Loch Ness is very big, and the sampling issue is a real one. So there will be limitations on our study, and so some of the limitations are maybe we didn't sample extensively enough, maybe we didn't sample in locations where these creatures reside, so that the DNA signal is reasonably localized within the loch, so they have to have been in proximity to that water for us to be able to get a sample. And of course, as I mentioned earlier, there's this temporal element. So if we were sampling in a place where the organism had been, but it was there a week before, then we would have missed it. And of course, when you make a announcement about the Loch Ness Monster, everybody's got a theory. And so, clearly I sampled at the wrong time. Nessie was on holiday, maybe visiting cousins in the south of France. And of course, my personal favourite is that Nessie is some form of mythical creature or extraterrestrial creature, and so why would I expect it to have DNA? To which I answered, well, I didn't find any unicorns either. But we started with the premise that this thing could have a biological reality, and so we tested the biology, and that's what we did. And we didn't find any evidence of anything biologically unusual that would explain the Loch Ness Monster, except for perhaps the eels. And the eels is interesting. So as soon as you get into sort of the Loch Ness mythology, people email you stuff. And some of the stuff that I received was quite interesting. So there were a number of people who claimed that they had seen giant eels. Okay, so that's interesting. There was a Canadian couple that reckon they saw something that was 20 feet long in an eel. I think that seems highly unlikely, but there are conger eels that can get to that sort of length. And there's also these stories, particularly from Foyers, which is on let me see if I can get this right, the west coast of Loch Ness, where there is a power station, and there are a number of people from the local region who have told me that they were at this power station, or they had friends who were at the power station in the 80s, and it was getting clogged up with, drumroll, giant eels. And it shut the entire power station down, and these eels were as thick as people's legs, and some claimed that they were, 10 to 15 feet in length. And so scientist Professor Neil Gemmell says, well, did anyone take a sample, or are there any photos of these things, or has it gone to the local museum? Because I thought that sounded pretty extraordinary, and the answer was no, it was just what happened. It was just normal. Nobody thought it was anything to get excited about. Which again, skeptic in me goes, I don't care if I was a power worker. If I got a 15-foot eel in my machinery, I'd think that was kind of interesting.
10 a penny in Scotland.
Yeah, 10 a penny in Scotland. I mean, it's like, obviously in the Highlands, it takes a lot to get people, riled or excited. Oh, 15-foot eel? Oh, I saw a 20-foot eel the other day. I mean, what the hell,
Yeah, the bar's pretty high.
Yeah, just, very, very late, very understated, you Scots. So
when you get that much liquid gold floating about the lochs,
Yeah. Anyway, so I don't know about the eel story, but there are a number of anecdotal stories that sort of support that. So anyway, I thought it was intriguing, but actually more interesting to me, and it's actually taken us a lot longer, is all the other biology that's present in Loch Ness. So Loch Ness is actually a really neat place, because it's so big and it's, so dramatic in the Great Glen, it's actually like a natural concentrator of the environmental signal from the entire region. So you find evidence of other species that might be of interest, things from the terrestrial environment, whether they be deer or sheep or goats or endangered species, some of the birds and mammals that are now less common than they used to be. So that was interesting. And then, of course, there's all the bacterial life and nematodes and various other things, because remembering that Loch Ness is very deep and it's very dark. So interestingly, at about 10 metres, light's almost completely knocked out and it's pitch black. And so you're getting into a very chemically driven ecology down in the depths of the loch. And we found a few bacteria that are kind of unusual. There's some that are very high salt tolerant bacteria, and I don't fully understand those, I'll be honest, but we're trying to work that out. And there's other things that might be involved in breaking down methane and other sorts of products. So in terms of the biology of the place, it's a bit more complicated than at first glance. But, in terms of vertebrate life, other than those fish, there wasn't too much there. There was certainly no reptilian DNA of any kind, not even an adder. In our samples, there were some toads and some newts, so some amphibians. There were all the fish species that had been recorded from Loch Ness over the years, right down to sort of minnows and sticklebacks and that sort of thing. Yeah, but no giant.
And was there anything? Were there any sequences that weren't known to science?
Well, there are about something like a fifth or a quarter of our data we couldn't easily put onto the Tree of Life. And so what happens is that your Nessie enthusiasts go, aha, that's where she is, or she, he, they. Nessie is in those sequences that you have just discarded, Professor Gemmell, because you're too lazy to look at them in detail. And the answer is actually we did look at those in some detail because we knew exactly that people would start going, well, there she is. Those sequences are generally short fragments. Sometimes they are incomplete sequences, so we can't map them as accurately to our databases as we would like, so we discard them. So they're partial products of the sequencing or of PCR. Sometimes they're PCR errors. So one of the things that happens during PCR is that sometimes you get mispriming and you get these false products. One of which is particularly problematic is a thing called primer-dimer, where the two primers actually bind to each other and make lots and lots of product. So that happens sometimes where you don't have very much DNA present and the primers can sort of interact with themselves in preference to anything else. We also had some what are called jumping products. So these are things that we know were probably started sequencing one piece of DNA and then somehow it became an amalgam of two sequences. So they happen very, very rarely, but again, and when you're doing lots of PCRs, those sorts of things can happen. So most studies that do environmental DNA generally have a large proportion of sequences which, for lack of a better way of describing it, are complete rubbish. And again, that's why that replication is so important. Each PCR reaction is an independent experiment, if you like. And so what you're looking for is the consistency across those. Anything that occurs just in one PCR on one occasion, I would be very dubious about. I'm not likely to believe it. If I find it in 20 samples with quite high abundance, I'm going to probably believe that that's a real thing. But a one-off hit or one sequence in one tube out of all this sort of work is probably an error.
We're leaving the door slightly open then.
Well, the door is open for people who want to believe, because there's always a way to explain it. Some of the explanations get more extraordinary. We gave it our best shot. We went in with an open mind. We didn't know what we were going to find. We didn't find any evidence that there is an extraordinary beast in Loch Ness, but there is enough doubt in our study, as there have been in every other study that has been undertaken at Loch Ness, to leave the door open. I mean, some people have gone with an expectation to find something, and at the end they have led to sometimes some extraordinary hoaxes and sometimes some extraordinary foolishness to try to find an explanation. for something that probably wasn't there, and probably, we've taken a slightly different approach. We've said, okay, we don't think there's anything there, but of course we could still be wrong. Every scientific study that's been undertaken at Loch Ness has come up empty for evidence of the monster, and we joined that. We joined that club, and as the technology gets better and better, people can go back and do it again. I mean, we could do the study again quite easily, as could others now, and they might come up with a different result. But really, what we were trying to do is to get people switched on to the fact that they can study their environment in a completely new way, using DNA sequences present in these environmental samples, and it's extraordinarily powerful for quantifying and documenting the life in a particular place. And we did that, I think, very successfully. I think more people know about environmental DNA now than they did when we started. I was encouraged. I'm not a huge social media person, but I was encouraged when we were doing our study that there were a few tweets coming out. There was one lady who's an eDNA person on the west coast of the United States, and she was getting ready for her expedition. She was going off looking for turtles in the Pacific Ocean, and she had all her boxes and things on the pier, and a couple came up to her and said, "Oh, what are you doing?" And she explained that she was going off looking for turtles and using environmental DNA. And they said, "Oh, like the Loch Ness project." And I was like, and she was like, "Yes, that's right." And I was like, "Yeah, people understand what this was all about, I think."
So what comes next, Neil, for you? You've obviously you continue, I guess, to work on those sequences and in search of Nessie. But can you tell us what's coming up for yourself and using that technology? Do you have plans to go somewhere else?
Yeah, so I think it's going to be hard to top Loch Ness for the publicity that we managed to generate from that project. I mean, if I have one disappointment, it's that the sort of the press component of that preceded the science. But I sort of released what we had found that was of public interest in September last year, partly because I kept getting emails saying, "You said you were going to give us the answer in January 2019," and of course I hadn't done that. And as the emails came through, I thought, oh, well, we might as well just get the stuff that people care about out. Normally, as a scientist, you like to have your scientific paper out and then you talk about it. In this case, we actually put some of the data out up front, and the paper, still to my shame, is not finished. It's on my to-do list and very, very high on that to-do list, and I've got probably 3/4 of it written, but I've been dragging the chain on that, as they say here. What's coming next? eDNA continues to be incredibly exciting. Off the back of Loch Ness and the fact that, for example, we found lots of terrestrial mammals and other species in Loch Ness, we started wondering about the power of this technology to monitor environments in a sort of total way. And so in New Zealand, we're very excited about the idea of using this technology to monitor our endangered species. So maybe from water samples and from rivers running through major national parks, we can get a very good census of not only the native species that are present in that area, and in some instances, some of our native species are so rare, it's very hard to get an accurate count on where they are and how many there are. So maybe we could start to get to a place where we can at least document where they are. Quantifying with environmental DNA is very hard. We're also interested in whether we can use that to detect what we call pest species here in New Zealand. So these are introduced mammalian predators. New Zealand had only one terrestrial mammal, or three terrestrial mammal species, one terrestrial mammal, three different types of bats when humans first arrived here. And of course, we've modified that quite substantially. So we've got rats, and we've got stoats, and we've got possums from Australia, and there are a number of other pest species that cause huge damage to our forests and to our wildlife. So we'd like to be able to monitor those. And we're also getting really excited about the idea that we might be able to find ways to monitor our environment using organisms that are already present there. So all the technology I was telling you about really distills down to collecting water and filtering it through laboratory filters. And that's quite time-consuming and expensive. And so one of the things we've been wondering is, are there quicker ways to do that? Either collecting larger sums of water using simpler technology over time, or maybe you could use the organisms that actually live in the water, things like sponges and mussels and other critters that are constantly filtering the water and concentrating that signal for you in their guts, and maybe we can use that as a way to monitor our environment. So those are two of our sort of hot topics, and we're not the only ones in the world that are working on this. So we've got colleagues in the UK who are investigating sponges as natural concentrators or natural samplers of environmental DNA, and there are many others that are using remote technologies. There's some really cool underwater vehicles now that cruise around environments and collect samples of environmental DNA for over a period of a week or a month. So I think the field is moving forward very rapidly with new technologies and new approaches, but the fundamental questions that can be answered remain the same. You know, what's in our environment, how much of that species is present, and most importantly, if we can do this repeatedly over time, and this is a much cheaper way than going out collecting things in boats or diving, although perhaps not as much fun, we can get very accurate censuses of life over time, and we can monitor how environments change. And that was another key message.
And of course, if you look at how much this whole area has progressed over the last 30 years, you know, the idea of where it could go in the coming 30 years is quite staggering. And of course, for the young people listening, they'll be coming up through that and you know, and developing with all that technology.
Yeah, I predict that in a few years' time there will be a device. There are already devices you can attach to your phone and do sequencing, but I would predict there'll be devices you can attach to your phone and do a full environmental DNA analysis and get the readout there and then. And I think that's exciting, not just as a scientist, but as a citizen who's interested in the environment. So, you know, there's lots of people who are interested in doing bird watching and various other things. Maybe environmental DNA will be a sort of a new hobby, a new way to monitor and look at things, whether it be your local community pond or your river or your oceans. Or just for understanding your own safety. You know, we're living in an unprecedented time of a pandemic. We need technologies where we can protect ourselves using sequencing or DNA detection in real time to know whether or not we're being exposed to things that might affect our health. And I think literally we're on the cusp of a transformation in how portable, accurate detection technologies will be commodified for citizen science type initiatives, and all those data will go up into some sort of cloud, and you'll be able to analyse them in real time. What's happening in particular places, whether it be about biodiversity or about public health. So that's again probably another chapter, but we're involved with other people around the world in using sewage samples now to monitor for COVID-19, and that work would have never happened if it hadn't been for environmental DNA and probably the Loch Ness project.
Fantastic. It really just leaves me to say a huge thank you, Neil, for coming on and speaking to everyone. It is, of course, the middle of the night here, but as we speak and the start of your day, so I'm conscious to let you get on. But thank you so much for giving your time and for speaking to us all. And, you know, hopefully at some point you can come back to Scotland and just double check that we've not missed anything up in Loch Ness.
Yeah, I would very much welcome that opportunity. I love Scotland very much. So you know, my mum's Scottish, so I have family and connection to there, which is actually probably part of the reason why I ended up going up to Loch Ness. But there we are.